So thank you very much, Neil. I'm very happy to be here to talk to you today, and I'm happy that you're on the panel because you're the one who asks all the both funny but also the very pertinent questions. So you're the perfect person to chair this. Before I start my talk, I'm trying out this new polling system that Sigoni introduced me to. So if you go to Slido, so sli.do, and then you use the data dialogue as the hashtag, you can participate in this poll. So you can say, where do you go when you find data for your research? What do you do? So you can all go, you can do it on your phone, you can do it on your laptop, whatever, if you have, there's probably a couple of you in the audience who don't have the internet access immediately, but those who do, you can participate in this poll to say where you go to find data for your research. I'm just giving you a couple of seconds so people can type through. Well, and you can see it change in real time. Ooh.
That'd be really awkward and ask whether Google means Google or Google Scholar.
Google means internet search, I guess.
Yeah. Google Scholar
Google Scholar, I think that would fall under a tool for data discovery, I think,
right.
or even literature search, because if it's really searching articles, then I would say literature search, where Google is more like a general web search. So it looks like most people have looked down on their electronic internet-connected device and now looked up again. So I guess that means that we now have at least a 30-something, there's still a couple of votes coming in. But really interesting, there's a, okay, it's still moving. Maybe you should leave it in the back while I continue on my presentation. So the last couple of people, they can come in. Oh gosh, it's still moving. But there's an overwhelming, two topics or two answers that come up top: literature search and Google for data. So, which is in one way, it's for me, it's reaffirming results that we found in the past by interviewing researchers. But it's also a bit troubling because if you know the amount of body of literature that's out there and the amount of web pages that are on the internet, and the type of data that you're after and the specificity of the data that you're after, these two options may not be actually good ways of finding the data that you need. So it's just a way of pointing out that there are lots of ways of getting to data, and it's interesting that the most common way of getting to data is through literature search, which is not necessarily an easy way to get to data. But we'll leave it up there. You can still go in and put your answers. Let's see if I can change this round to my presentation. So in the, it's this one. Yeah, you just need to scroll up to the top. It's this one. Yeah. So in the presentations we had so far, there's been a lot of talk about data sharing, and data sharing is actually a bit of a problematic term because data sharing includes quite a lot of different things. Well, first of all, it includes some data being produced somewhere. Then it includes that data being stored somewhere. It may be stored several places, but then it includes someone making that data accessible or visible for others to see. Then it includes someone else finding that data. Then it includes those people gaining access to that data. And all of those steps is all included in what we call data sharing. So all of those steps in themselves have their own difficulties of various things that are problematic. So where do you store the data in the first place if you have data files that are enormous, which was it Henry who was mentioning that was the reason why Mark Hennell started Figshare, was because he just had really large data files, and if you don't have the infrastructure to store really large data files, what then do you do? And then you get something like CERN coming up, and they have even larger data files. They need somewhere to store it. What do? They create a repository to host that particular type of data. So storage in itself is a problem. But then comes the whole thing about enabling access to data, which may have things about sensitivity. It may have things about consent for what you can use the data for, especially if it's personal data or clinical data. And then on the other side, you have all the activities of then gaining access to the data. And the topic that I chose to do in my talk, and I know I've spent a lot of the time just introducing, but is on the data discoverability side. So I work with, as Neil mentioned, DNA Digest, which is a registered charity in the UK, and Repositive, which is a social enterprise, where we help genetic researchers find and access data. And it sounds so simple that it almost sounds stupid, like, well, can't they find the data themselves? They got it, they must know where it is. But it's not that simple. And I'm trying to give you a couple of reasons why. So you might have heard of, I mean, especially if you live here around Cambridge, of all the genomic technologies and DNA sequencing, which is becoming ever cheaper, which has the result and the effect that data is produced ever faster with even higher throughput technologies. So the amount of data that's coming out of these new technologies was forecasted to have been, and this is only looking at human genomics data, more than 80 petabyte of human genomics data sequenced per year was the capacity of data that could be sequenced. And that was last year. And as you can see, this curve is going up exponentially. So I dare hardly guess what the what the estimate is for this year. But then if you compare to what data is actually available in repositories that are storing human genomic data, the total, so what is gathered over all time to date, is less than half a petabyte, right? So if this is the annual capacity, and what is actually available for research and reuse is less than half a petabyte. So it's a tiny percentage of this. So there's all this data being produced, and there's lots of researchers that want to get to it, but first problem is it's not all accessible. So it's not necessarily in a place where you can access it in a public repository where you can find it. There's even more data being produced as a result of the data production being cheap. So you have population sequencing projects turning up all over the world. So you have, for example, the 100,000 Genomes Project in the UK. You have another 100,000 Genomes Project in Saudi Arabia. You have the Icelandic population that was sequenced in its entirety. You have the Veterans Program in the US, with another 100,000 genomes, and health record, etc., and it goes on and on and on. So because the technology is enabling, that means even more data is being produced, and it's being produced all over the world in many different contexts, in many different projects. Now, the individual researcher, and here we're talking about human genomics, when they go to look for data, they think, okay, where can I find data? And then they can usually mention 4 to 5 public repositories that they know hold some form of human genomics data. That is okay, except from already in this incomplete map I just showed you, you can see there's more than 4 to 5 flags on the map. And what we found is that there's hundreds of data sources out there, and any one of those could be the one that has the type of data that you're interested in for your research. Okay, so you can't see this, but I put a link at the bottom of the slide. So the slides are shared online afterwards, so you can go check if you want to see a list of data sources. But just to give some examples, there's lots of data sources that have been established either within individual institutions or within individual research projects, some of them specific for certain diseases, so for example ADHD or autism, or it could be for specific types of data. So for example, there's specific repositories for methylation data or for longitudinal data, specific repositories for cell lines. But you also have different assay types. So depending on what type of genomic data, is it sequencing data, is it array data, is it ChIP-seq data, you might have different repositories that have them, or you have repositories that have many different data types. But already this shows that you as an individual researcher, where do you go to find the type of data that you're after? Well, either you need to already have a memory like, I don't know if it's elephants, I don't know if they remember repositories, but but you need to know of all these different data sources, not just know of them, but know which data type you would find in each of them, which is quite a stretch. And the amount of data that's available in them ranges from anything from tens of samples to millions of samples, which, for example, one of the very big ones is, for example, a big biobank in Finland that has more than 1 million samples of genomic information from the population studies. And this is, the data is also representing populations from all over the world. So I have a bit of stats here just to show where the data comes from and who's funding it. So you can look more into this on the slides, but just so we save some time. But the first thing is that all those data sources, that's already just a tiny fraction of all the data that's already out there. So there's all the data that's not there yet. So it's not even accessible. And what is the reason for this? So there's already been mentioned some very excellent talks this morning and also just now about the cultural barriers for data sharing. So there might be a wish to keep all your results for yourself until you've published everything that you could ever publish or find in that data before you share it with someone else. Or it could be if it's often when it's more commercially minded, or actually also in academia, it can be a lot of competition. I need to do it before my neighbor, so that's why I keep it to myself and before the neighbor sees it. But in the type of research studies like genomics, where any data that you have is never enough, because in genomics it's a numbers game. The genome is big. The statistics say that you need tens of thousands of samples before anything is validated. So whatever you find in the data you have, you need more data and even more data in order to validate it. Otherwise, it's much more likely to just be a bias in your sampling or a bias in your methods. So you need lots of data to validate. So there's the huge need for actually opening up these silos and sharing the data, making it available. But there's additional challenges, such as when it's genomic data, you probably know that all of you have your own unique DNA genome. So each of you, although you're very alike as humans, you have very similar genomes, all of you have unique identifiers in your DNA. So if you share the data for your DNA, that's identifiable for you as an individual. So even if you remove things like your name and birth date and address, if you share your whole genome, that could be used to identify you uniquely among the whole population of the world. So suddenly you have questions about the data privacy, which is specific for this type of data. How do you then share that data with other researchers without compromising the privacy of the patients and their respecting their what consent they have given? Because if they do not want to be identified, how do you then make sure that you can still use the data for research without identifying the individuals? So that includes everything that has to do with data governance and ensuring that there's sufficient patient consent given, and then whatever legislation is around on how you can then use that data for medical research. And all of these become barriers. And I know there's, I mean, I can talk for ages about this, but I just want to focus on the discoverability issues. So there's the counter movement to this, because you can think like, okay, with all these problems, and all the researchers would probably not share data at all because it's just too difficult, and they all run out of time, as we just heard. You're at the end of your grant, you need to write the next grant, and you don't want to spend the time getting through all these barriers for sharing. But then at the same time, there's a push from the other side, for example, community-led resources. So there's a number of with the rise of direct-to-consumer genetic testing, individuals, either healthy individuals or patients that have access to their own data and choose to share it openly in order to make it available to the research community. So you get these communities of, it can either be patient groups, or it can be open groups or resources on the internet, where individuals have chosen to share the data openly for research, for the common good. And when it's the individuals choosing to share it, then you suddenly beyond the whole problem of the patient consent and how you get through the custodianship role that normally the university of the researchers otherwise take on them. But of course, this is not a solution for all. And if you're interested in hearing about these, I can tell you about a number of these projects, like the Personal Genomes Project, OpenSNP, Genomes Unzipped, and there's a number of these. I'm happy to tell you more about those if you come up to me afterwards. So there's something that needs to change, because if it's all so difficult to share the data, but there is data out there, but it's difficult to find it, how do you then make the most of it? Because if you don't get access to it and if you don't share it, well, how are you ever going to find those targets for the next drug, which will help cure the cancer of your mother tomorrow, right? So that the changes that needs to happen in the data sharing is not just for the benefit of the researchers, it's actually for the benefit of the patients, it's benefit of who will be the patients tomorrow and who have the interest in actually the research results in the end. So it's very important to step outside the view of just the individual researcher who's thinking more about grants and funding than about the patient's interest, but think about the interest of the patients who donated their data to research in the first place. So if we summarize the type of pain points that's, and this is specifically related to the discoverability and access to data, part of it is because the data is fragmented, it's in many different places, so it's difficult for you to find where the data is. There's a huge admin burden to manage data access, and that's both for the individual researcher who is looking to access data, but also for the repository who's managing the access. How do you manage that relationship? How do you check that this researcher is actually using the data in the way that they're supposed to, that adheres to the patient consent, etc.? And it's such a painful process that process alone makes people not want to share data, not want to access data. And then there's this what I call bad culture. So I didn't have another word to put on it, but it's more of a habit or a remnant of the time when research was something that you could just do at home in your lab or your own little lab, and then when you came with the result you can show the world. Well, the kind of research that we're doing today, especially in genomics and other big data fields, is dependent on the data access. So you can't get to the results that you want without having access to the data. So you must collaborate, you must share, and you must make your data accessible, because otherwise the whole field is not going to move to where we want it to be, and everybody wants personalized medicine that will cure them faster than yesterday. So part of the best practices to overcome these things is, first of all, to make data discoverable, and that's part of the work that we're doing at Repositive, because we're making all the data visible to the researchers. But there's also a big part of simplifying the workflows, and that goes again for both sides of the equation, both for the workflows for the repository to make, or researchers to make data available, but also for researchers that are trying to access data. And then there's something about the sense of community and understanding those incentives, because if just every researcher, if every grad student knew both at the start and the end of their studies all the 5 selfish reasons to do reproducible research and do open science and to share data with the community, if they all had that top front and center in the work that they were doing, it would be part of a community exercise and be part of the contributions that would be recognized and incentivized in all the work that we're doing. I know that there's a number of things that have to change for that to happen and be part of the natural research culture, but that's what I would like to see, and I hope people in this room follow me in that thought, so you can also advocate that for your researcher friends and the young students. And again, I put a link at the bottom of this slide, which talks about how we are working on a couple of these things through the work of DNA Digest and Repositive. So just a short slide. I don't know how much time I have left. Okay, thanks. Okay, so with the Repositive platform, we have made, and I found the discussion that we had in the earlier session interesting about, so what does it mean that one of the services you're using is a commercial service, because Repositive is a commercial company. It's a social enterprise, but it's a commercial company. Commercial just means that we need to find a way to recover costs in a way that the company is profitable, because otherwise the company won't exist tomorrow. So it's like a very fair reason, but how do you combine that with making something that is contributing in the best way to the community? So we've found a way to make a business model in which we, as part of the work we do, part of that work includes making a portal where there's free access to search for data for researchers. So we're providing that as a free service to the community, but then we're providing paid services on top of that. So part of the free service is that we are indexing all the data sources around the world. So that's indexing public repositories, it's indexing private repositories, data in biobanks, data in commercial companies, all data that has been consented for research use, and researchers can either access directly or they can apply for access to it. They can search all of those through one portal through the Repositive platform, and that's free to use and available for anyone. You can use it to find what data is available, see the can't see this one anymore. You can see the data descriptions, and you just click through to access. Because it's a platform where we keep track of the users. That means that we can vet the users, so we know whether this is someone from a university or this is someone from a company, so that you know who you're talking to on the platform, which helps build the trust in the community. So if you know someone is commenting on a dataset, this is actually a real researcher commenting on a dataset. It's not a random person from the internet. There's like this sort of level of difference. If you know who the users are, then suddenly there's a different types of communication going on. And the whole idea with the Repositive platform is that instead of being just static data descriptions, the community can help each other by add comments and feedback, discuss data sets to say this data set was useful, this data set was missing some information, this data set was used in this study, and so on. And in that way build the usefulness of the data sets over time, so they don't just become a static resource that everyone forgets about. So I know that the program says that there's a panel discussion later on, so I thought I'd just put in a couple of things that I'd like to talk about. Everybody already mentioned fair data. I put that here as well. So we could talk some more about that, but we could also talk more about what are the incentives in the culture of researchers that need to change, and what are the habits that we should help teach the students early on, that these are the best ways of working with data, these are the best ways of working with your colleagues, so that you benefit not just your own research, but also the whole research community. And I'm happy to talk more about that. And then I promised the people from the Wellcome Trust to put in an extra slide to advertise that they have this course coming up in December, where I'm on the program committee, and we have one day of the course is about commercializing data in genomics and discussing what are actually the business models that work. So anyone who's interested in that, or in either in genomics, or there's somehow in translating research, technology transfer offices or similar, go to the Genome campus in December, because it's going to be a really exciting course. So with that, I'll say thank you and take any questions. Any questions?
Any younger folk with questions?
Older people welcome too.
Okay, so we have a question here.
One of your problems.
Oh,
No. The chap behind you had a hand up too, so we'll do that too.
you look very young, sir. Go ahead.
Thank you. You've rendered me speechless for that comment. You talked about the problems of data being distributed, completely sort of scattered all over the world. But what about metadata? Should that be centralized? I mean, if you have centralized metadata, doesn't that solve the problem of fragmented data?
Yeah, it would. So in a sense, that's what we're doing, because it's by indexing data, we're talking about indexing metadata. So we're not collecting data, we're just indexing the metadata, which means that you can search it through one portal. So behind the scenes, that means that the metadata is stored and indexed in a common framework.
For yourselves, but what about around the world? For everybody?
No. So that's the public resource. That's for everybody. Right. Yeah,
Okay.
exactly. Because that's exactly the point. Because if the metadata is scattered all over, you don't know where to search. Because if you search here, you only search this part of it. If you search here, you only search this part of it.
I made the case for DataSite being one centralized metadata repository, but are there many fragmented data sites around the world, or is DataSite pretty much the accepted place for centralization?
So the uncomfortable truth is that there's many different ways of making data available, and what we found is that I didn't actually include the stats here, but the number of institutional repositories that's just been set up just because we have to, they range anything from somebody setting up an SQL database to someone just starting a website and making links to PDF documents of the data, of the publications that mentions the data that you could access. So anything there and in between, it's not following a standardized format and not necessarily issuing DOIs. And even the metadata formats, there's no standard at all. So even though there is standard, I mean there are standards and there are centralized resources where everything is standardized, anything that just pops up tends to not be part of those indices, and it just becomes invisible, because then you just use your most common portal or whatever. Maybe you're just searching DataSite. Maybe that's your portal of choice for finding data. But that doesn't necessarily mean that you see all the data that's available. So that's a problem. So we overcome that by just doing the hard work in the back of indexing also everything that doesn't have DOIs and have ugly formats. But it's of course just a way of overcoming the problem. It doesn't stop new repositories from popping up that don't adhere to the standard, if you see what I mean. So addressing that separately of how do you make any new repository that pops up actually use existing standards, I don't know how, but about information sharing, telling them that these standards exist and these are the benefits of those standards.
What you need is metadata.
Right, so we had another question there, and then we'll move on.
So I have a question and a comment. So my first question is that I'm a stem cell biologist, and whenever we publish a paper, we automatically submit all our metadata to the journals, and then it becomes available by supplementary data and all that kind of stuff. So how is that different from Repositive? So it's access to anyone who has then obviously access to the journal if it's a closed journal, but if it's an open journal, then it's anyone who has that access can access it. So that's my first question. My second comment is, what is your ideas about? You're saying like people have the personal option of making their genomic data available for research, but a lot of the times we see that going into I think there's a recent company in the U.S. who sold 70 genomic sequence data to a private company for I think 70 million, so they can have only access to that. I think it was Parkinson's disease genomic data. So how is that different from?
So two questions, right? So the
Can
first
we
one.
add the first question to the panel discussion? Because I think that's basically why would you trust a journal is the answer.
Okay.
But there's the second question.
And the second question, I think you're referring to 23andMe that has a huge collection of data, including data for some diseases like Alzheimer's, and they made a sale of access to that data to a pharmaceutical company. So the way they've collected their data, so the way they made their data collection was by selling these very cheap direct-to-consumer tests where users can get answers to some questions like what eye color am I likely to have? Things like this, and some things about ancestry and some more interesting things as well. But as part of that, they get access to download their own data, but also as part of the process gave 23andMe consent for using their data for research purposes. So those who did not notice that small print when they took the test might be disappointed, like why are they selling my data? But actually it was part of the contract that you made when you bought this very, very cheap genetic test from them. Yeah. So it was part of the consent of that particular data. Yeah.